to introduce you to this final pre-Christmas session of the liver cancer program. We're having a great audience, and that basically speaks, for you, being the reason for this. So let me just briefly introduce for the two people on the line that don't know who Scott Friedman is. He is a Dean for Therapeutics Discovery and Chief of Liver Disease at Icahn School of Medicine at Mount Sinai. And, he is clearly an expert in hepatology, more specifically liver fibrosis, chronic liver diseases, and somebody who has spent a lot of time covering everything from basic research and understanding mechanisms to translational studies and clinical trials in this field. He initially graduated from Icahn School of Medicine in Mount Sinai, then was a resident at the Beth Israel Hospital, Harvard Medical School, went to UCSF, at a senior Fulbright at the Weizmann Institute, and since then has been very, very successful. And one of this is mirrored by more than 85 fellows and students that went through his lab. And obviously he has been awarded many awards, including the EASL International Recognition Award and Distinguished Achievement Award from AASLD and the American Liver Foundation. And without further ado, Scott, very pleased to have you speak. And but last but not least, I need to mention he's obviously also on our advisory board actually for the liver program.
Which is a big honor. Thank you, Tim. Thank you, Anu. Thanks, everybody. I see a lot of friends. So as I mentioned before, it makes me a little nervous, but I'll do my best to keep you entertained and maybe even learn a thing or two. So let me just make sure. Can you see the slides advanced?
Yes.
Perfect. Okay, these are my disclosures. Rather than run away from them, I'll say I'm proud of the fact that what we're doing has translational potential and let you know that I'm very optimistic about the prospects for new therapies for liver fibrosis. These are the three main areas I'm going to cover in the next 45 minutes or so, and I should leave enough time for questions. So, but if you have burning questions, feel free to raise your hand. I think I'll see it, but certainly at the end. I want to provide a little clinical context for hepatocellular carcinoma and metabolic dysfunction associated steatotic liver disease. I know it's a mouthful. For those who haven't been at the recent liver meetings, this is the new name of what was previously called NASH. And the equivalent of NAFLD is now MASLD, and I'll point that out in a slide in a second. As many of you know, my longstanding interest has actually been in the biology of hepatic stellate cells, which are the liver's pericyte and the primary source of fibrogenic myofibroblasts. And I'll emphasize how much more we've learned about their heterogeneity, and with it, evolving therapeutic targets for different stages of disease. And then hint at some of the work that's been done and is ongoing for cell therapy of fibrosis, targeting in particular senescent stellate cells. So I'm going to mostly restrict my comments, although not entirely, to this disease, which I mentioned already is now called MASLD. It's a progression from plain steatosis to an inflammatory necrotic disease that is accumulating fibrosis over many years and ultimately culminates in cirrhosis in a sizable fraction of patients. Just to remind you again, this is the equivalent name change. And traditionally, thanks in large measure to David Kleiner and Beth Brunt, a scoring system was evolved to measure the different components of the disease: steatosis, lobular inflammation, and ballooning, and fibrosis is scored separately. And I think we've all learned that while these different markers or stages are informative, they do not correlate with outcomes. The only thing on this list that does is fibrosis. We also know, of course, that hepatocellular carcinoma can eventuate, and I emphasize here on the right-hand side that there are more patients with hepatocellular carcinoma who are non-cirrhotic in NASH or MASH. I'm going to make that mistake all afternoon, so bear with me. Patients with MASH, as I'll show you in the next slide, have a higher propensity to develop carcinoma without being cirrhotic, although they typically have advanced fibrosis. Just one other point. It's from a nice review from Zobair-Yanasi to underscore that the rate of rise of the incidence of hepatocellular carcinoma is greatest for MASH than for all other etiologies, although it still comprises a smaller fraction of the total number of cases. the bulk of which worldwide are attributable to hepatitis B and C. There is, speaking of worldwide, a marked geographic distribution. Some of the data we don't have. The darker the color represents the higher the incidence of NASH, HCC. This is from a review that I co-wrote with Josep and several other authors, Josep Lovet. And clearly there are regions that are real hotspots, the Middle East and North Africa, the obvious one because of the prevalence of, or until recently, the prevalence of hepatitis C, but also Mongolia because of the prevalence of co-infection of hep C and hep B, high alcohol use, and even some hepatitis delta. So this is a rising problem, obviously, with enormous clinical consequences, not the least of which is typically when the disease is diagnosed when patients are symptomatic rather than through screening, the outcomes are generally poor. The point I made a couple of slides ago is underscored by these data. This is from the VA hospital in Houston, and it emphasizes that across the different etiologies, whereas hepatitis C, B, and alcohol typically have 90+% of the patients with cirrhosis before cancer arises, there's a larger fraction of non-cirrhotic cancers of up to a third, and this has been held up in 2 or 3 different studies now, that suggests that in a MASLD or MASH, the microenvironment, as we well know, and there are many experts on the call who know more, or on the lecture that know more than I do, but it is pretty clear in a broad sense that the microenvironment in steatotic liver disease has many unique features, not the least of which is that patients are typically obese, and they have all the drivers of cancer that heighten the risk of cancer of all types associated with obesity. But this remains an important issue, and while screening guidelines do not yet recognize or emphasize the need to screen before patients are cirrhotic, I think it's prudent among clinicians that if your patient has advanced fibrosis, not to wait until they're cirrhotic before one starts screening. The outcome of waiting is that typically HCCs that crop up in patients with steatotic liver disease tend to be larger and less amenable to curative therapies, only because they're not being screened. To return to the concept of fibrosis as a risk factor, this has been known through different studies for at least 15 years, but probably the best and most definitive was published in the New England Journal a couple of years ago. Arun Sanyal led this, and of course it was the extraordinarily successful NIDDK NASH CRN, led by folks here on this call, in which the group followed 1,773 patients for 10 years and looked at the outcomes over that 10-year period based on the initial biopsy score at the time of enrollment. And death from any cause was certainly highest in the F4, followed by F3. A striking hazard ratio for hepatic decompensation, up to 52-fold hazard ratio for F4 versus F02, and 18.3 for F3 versus F02. So clearly hepatic decompensation was a marked readout of advanced fibrosis. And so is hepatocellular carcinoma, although interestingly, the greater risk appeared to be F3. And I'd love to speak to Arun. I haven't actually asked him why this discrepancy may be related to the number of cases, but if anyone on the call knows, please chime in now or at the end of the lecture. So one question as we try to develop antifibrotics is, what difference does it make if we regress fibrosis? How do we know that's going to leave patients with a better prognosis? We don't have any definitive prospective data per se, but what we do have is these data from 2 Gilead trials that failed. The data were organized by Arun Sanyal and others. There were 1,135 patients in aggregate. And although overall these drugs failed, one was selonsertib, the other was a LOXL2 inhibitor. But the point is that overall the drugs failed, yet there were 16% of patients who had fibrosis regression. Unclear why. It could have been that there's a subgroup of patients who were responsive to this meds. We don't really know, but nonetheless, one could ask what happens to those patients who had bona fide regression on biopsy. So if you look at the liver-related events in the patients who did not improve or have regression, here it is based on their NASH CRN fibrosis score, and here it is based on their Ishak score. But look at the marked difference in the likelihood of liver-related events in those small group of patients who had bona fide regression. So in an indirect way, this at least tells us that there is some value and rationale to seek to reduce fibrosis in all chronic liver diseases, but certainly in MASLD. Now let me turn to more of the meat of the lecture. That was really for those who don't think about this disease much, to provide some clinical context. Again, my interest has been in stellate cell biology, and I'm going to underscore several of these concepts. So this is a slide that's now almost 22 years old. I had it drawn for a review in an obscure journal along with my co-author and friend Michael Arthur. And it still holds up very well. This is to depict the anatomic orientation of the sinusoid with respect to where stellate cells are. So stellate cells are a pericyte-like cell. They surround the sinusoid. They are akin to renal pericytes and probably similar cells in lung and many tissues. What's unique about stellate cells that we recognized early on is that they have these perinuclear droplets that store vitamin A. And in fact, this cell type is the primary storage site for retinoids in the body. And we also learned that as the liver gets injured, these cells undergo a transformation histologically, which we called myofibroblasts. They lose their vitamin A, they accumulate extracellular matrix in this subendothelial space. So this is really the famous capillarization that was described by Popper and Schaffner over 50 years ago. And this represents the earliest signs or indication of fibrosis accumulation. And yet even here, may lead to dysregulation of surrounding cells, and particularly loss of differentiated function of hepatocytes and loss of fenestrae or pores. And so this has been a general paradigm that stood the test of time now for, in my experience, in my career, over 40 years. But of course, the story has become increasingly nuanced, as you'll learn. So from that original general description of activation, we and many in the field were able to piece together a model that suggests that first what happens is stellate cells have to be activated. That confers a whole host of responses that are not true of the quiescent cell. But once these cells are activated, they are responsive to a new range of signals that drive proliferation, contractility, fibrogenesis, altered matrix degradation, chemotaxis, as well as immunomodulation and inflammatory signaling. So I always like to think of this as a conspiracy to make scar, because each one of these features in one way or the other contributes to the phenotype that we know very well as advanced fibrosis or cirrhosis. In more recent years, there's been wonderful studies by Robert Schwabe, Tatiana Kiseleva, John Iredale, who uncovered some of the pathways by which stellate cells are cleared when the liver injury is resolved and the liver goes from a fibrotic to a non-fibrotic state. And it's worth remembering, we have ample example of fibrosis regression after cure of Hep C in particular, or even suppression of Hep B, wherein even early cirrhosis can be regressible. It's an astounding response, and it's despite the fact that the only directed therapy is to the virus and not to any of the cells per se. So we know that cells can either undergo programmed cell death, deactivation to an intermediate state, which has been described by several groups now. And interestingly, also, there's a subgroup that becomes senescent, and I'll return to that in a minute. By the way, sorry about the siren, but I'm in the middle of Manhattan, so it's part of the scenery. I apologize. So before I get to the cells themselves, there are a number of ways that fibrosis can be, the scar itself can be linked to hepatocellular carcinoma in addition to the cells. Certainly, these disorders generate chronic inflammation. That can lead to myofibroblast activation and generate cirrhosis, fibrosis, and then cirrhosis. There's also, and I'm going to focus on this, which is why it's in a larger font, it's thought that HSC senescence may drive cancer. There are other drivers that I won't talk about today, except to mention that activated stellate cells are known to be pro-inflammatory, or at least some of them, and they can reduce immune surveillance and increase tolerance, which is great if you're giving immunosuppressives, but not great if you're trying to mount an immune response to a tumor. There are also physical consequences of fibrosis accumulation, in particular the stroma and the liver get stiff, and stiffness per se is a driver of cell replication, both for hepatocytes, but also for stellate cells. We're learning a lot about this. This is barely summarized in this line, which indicates that integrin and matrix signaling are part of that stiffness response. One of the more interesting receptors lately, particularly in the context of hepatocellular carcinoma, is DDR1. And for those interested, I encourage you to read more about it. There's a very interesting and somewhat complex contributions of DDR1 to the tumor-prone environment. And of course, the matrix is also a reservoir for growth factors, many of them proliferative, like HGF, insulin-like growth factor. And so as the matrix is laid down and perhaps remodeled, many of these growth factors can be released into the extracellular milieu and drive carcinoma development. The field of stellate cell biology was plodding along at a pretty nice clip, but has been revolutionized, like many fields, by the introduction of single cell analyses. This is the landmark paper of Prakash Ramachandran, who is in the group at Edinburgh, along with Neil Henderson, Stuart Forbes, John Fallowfield, a terrific group. And they really led the way in defining, using single cell sequencing, the landscape of cellular infiltration or composition in normal and cirrhotic human liver. They described, in particular with respect to stellate cells, 3 different populations: vascular smooth muscle cells that were mesenchymal-like, stellate cell, classic stellate cells, and then what they called scar-associated mesenchymal cells, which are more or less equivalent to myofibroblasts, and then finally mesothelial cells. And what they showed is that there's, of course, an evolution in the nature of the cell populations within the scar as cirrhosis develops. So the vascular smooth muscle mesenchymal compartment goes down, stellate cells stay the same, but there's a marked increase in the representation of scar-associated mesenchymal cells, and interestingly, mesothelial cells. And I'll skip this data and just point out that this leads to the concept of cancer-associated fibroblasts. And it's pretty clear, both from our data and many studies in the field, that cancer-associated fibroblasts, at least in parenchymal liver disease, are derived entirely or almost entirely from resident pericytes or stellate cells. And so stellate cells give rise to CAFs. And if you follow the field, whether in liver or pancreas, breast, or other cancers that are associated with a dense stroma, there's been tremendous controversy about whether these CAFs are pro or anti-tumorigenic. A hint to why this has been so confusing was uncovered by Robert Schwabe at Columbia in this landmark paper now almost a year and a half ago, in which they identified, or at least built on the concept that hepatic stellate cells are heterogeneous by assigning different subtypes of stellate cells to different and often opposing functions. In simplest form, they identified 2 major classes of stellate cells, one that they called cytokine stellate cells, which were HGF enriched and had anti-tumor activity, and COL1 enriched pro-tumor stellate cells that are called myofibroblast stellate cells. And this actually is a very, I would say, logical and in some ways comforting finding, because it does speak to the heterogeneity, and within that heterogeneity of stellate cells come remarkably diametrically opposed functions. And so you can imagine that the relative balance, if these cells are plastic and can evolve in different ways, the relative balance of these different cell types could have an outsized impact on the likelihood of developing cancer. We recently, I'm sorry, I'm having trouble with my pointer. We recently wrote a review that was just appeared in Nature Reviews Gastro. The first author is a very talented Brazilian scientist who's working as a visiting scientist in my lab, Bruno Cogliatti. And we tried to create what is more of a theoretical than an actual framework for the heterogeneity of stellate cells in a sort of a progression where cells are basically inactivated or inflammatory. Sorry, these are inactivated, these are inflammatory. They can either be inflammatory stellate cells or CAFs. There are also vascular stellate cells that can evolve into vascular CAFs. There are antigen-presenting CAFs, and there are contractility and migratory CAFs that ultimately lead to the so-called myofibroblastic CAFs. Now, this is obviously an oversimplification. I don't think every one of these stages is obligate in progressing to the next or later stages. And my suspicion, although I can't tell you with certainty, is this goes in both directions. So it's very plastic and therefore very challenging to capture experimentally. So if we return to Robert Schwabe's lovely paper, these inflammatory CAFs are presumably comparable to his cytokine calves, which are anti-tumorigenic, compared to the myofibroblastic calves that are collagen-rich in secretion and pro-tumorigenic. Again, a very simplistic notion, but at least a working framework to think about how this heterogeneity of stellate cells and their evolution could lead to different states of disease. And finally, deactivation, I already pointed out in that earlier slide, has been studied now more intensively. There are a whole family of transcription factors that actually drive deactivation. The most notable is GATA4, LHX2, TCF21 are the three that are really most prominent. And one can confer a more inactivated phenotype by driving expression of these inactivating factors. So activation is not just a one-way street. There is an equally potent transcriptional program that can drive stellate cells away from activation towards deactivation or possibly senescence, as I'll return to. I'm not going to read this to you, but this is just a little bit more granularity on those different subtypes and what the literature tells us about the heterogeneity of these subtypes, both in human and mouse, what genes are regulated, and what the potential functional implications of that are. And I will say that depending on how one cuts the data, there are as many as 6 or 8 different subtypes of stellate cells. There was a lovely paper from Dave Brenner and his students, Susan Rosenthal, in hepatology a couple of years ago, where they described, I think, 6 or 8 different subclasses. One of the challenges is what constitutes a separate class, and there's no strict definition for that. But to me, the most important feature is how are these classifications translated into different functions, because ultimately that's what we want to interrogate. So let me return now to the concept of heterogeneity with a slightly different twist based on work that was performed in my lab by an incredibly talented postdoc, so talented, in fact, that I was able to convince our leadership to keep her on as faculty. And she just started her lab here at about 6 months ago, and her name is Sammy or Shuang Wang. And she underscored, or I should say uncovered, a new concept about heterogeneity, and I'll emphasize that it's rather than just organ or species specific, is the fact that the progression of fibrosis yields a progressively different set of signaling models that drive fibrogenesis. To do these studies, we've used a model that we developed that's similar to others that have been developed before, but we've really characterized this model in great detail. It was performed initially by a visiting scientist from Japan, Takuma Tsuchida. We call it the FAT-MASH, or now FAT-MASH model, FAT standing for fibrosis and tumors, because as we give high fat, high cholesterol, high fructose diet, we also supplement that with a very, very low dose of weekly CCL4, 1/30th of the dose that's usually used as a monotoxin to induce fibrosis. And what became clear is not only do the animals develop fibrosis, but they also develop cancers. And the cancers that develop are almost exclusively in the group that has both the Western diet and low dose CCL4. And so this has been a very robust model for us. Just recently, we also had a paper accepted in collaboration with our nephrology team that shows that the mice also develop advanced renal fibrosis. And so it indicates that in some ways this mimics the other extra hepatic features of the metabolic syndrome. This is beta-galactosidase staining, which looks like it's localized to stellate cells, and I'll get back to that in much more detail in just a few minutes. So if we now look at the FAT-MASH model over a 24-week period, what we see, of course, is progressive accumulation of scar. And using a digital platform from a company known as PharmaNest, we can do much better than just a crude pathologic scoring and actually characterize and quantify through AI or digital pathology rather, the progressive changes in all of these features of the scar, such that by 24 weeks, there's a marked difference in collagen, fibrosis, architectural changes, et cetera. And that's turned out to be a very sensitive and powerful tool, particularly in the experimental context in which we use it. Now, one of the things that Sammy did that really made this study happen was that she decided, it was actually a medical student, not even a medical student, she's now a medical student, a college student who was working with us for the summer, an exceptionally bright young woman named Eliana Pickholtz, had been reading, and she heard about glass imaging of brain and tissues. And she said, can we try it in liver? And I said, sure, why not? And so with Sammy's help, they developed this clearing method where you effectively emulsify and lose the density of the liver. You can see that because you can't read the text behind these pieces of liver before clearing, and after clearing, you can see that the text is now visible. So Sammy developed and really perfected this method in liver and was able to apply Desmond staining, which is probably the most ubiquitous marker of stellate cells. And what you can see here are these gorgeous pericytes with long foot processes staining with Desmond that are remarkably similar to the drawings that were made up to 50 and 100 years ago by the great pathologists of old, and also Kenjiro Wake, who is an esteemed Japanese pathologist now in his 80s. So it really speaks to this kind of foot processes and morphology of stellate cells that is quite characteristic and looks somewhat like neurons or axons, which is not a coincidence because stellate cells do express a lot of neural crest markers. So what Sammy then did is she applied tissue clearing in a quantitative way across the stages of the fat NASH model. And as you can see, going from left to right with progressive fibrosis, there was an increasingly dense meshwork or network that she could quantify using image analysis to define the number of cell-cell interactions among stellate cells over time. And so what you can see here is as the disease advances, there's a growing number of cells interacting based on the connection to specific nuclei. And this is all modeled here where there's presumably increased autocrine interaction. So this, of course, was complemented by single nuke sequencing. This is Sammy's work in which she defined the gene expression patterns or profiles of the quiescent stellate cells labeled HSC1 and the activated stellate cells labeled HSC2, and the predictable pathways were upregulated in the activated cells. But the story became particularly interesting when she crunched the data using a software platform known as Cellphone DB, which actually, based on the density of the transcripts, predicts cell-cell interactions. And in particular, this is depicted as you see here, where the cell types are labeled in green dots, and the red lines indicate the likelihood of cell-cell crosstalk, and the thickness of the red line represents the density or quantity of those cell-cell interactions. So what you can see here is between the activated stellate cells, there's some marked interactions with endothelial cells and with cholangiocytes, but the most intense interactions were with the stellate cells themselves, which, if you think about it, overlaps nicely with the imaging that I just showed you that shows more and more cell contacts. I will say that the normal mice don't have this kind of autocrine interaction. I'll show you that. This is the human data. Interesting and satisfyingly, it's a little less clean. This is the control normal liver. These are NASH livers, also analyzed by single nuke sequencing. Clearly, you still have a stellate cell autocrine loop that wasn't apparent in the normal liver. Here, however, you have also more interactions with different endothelial cell subtypes and cholangiocytes. In a way, this is exactly what one would expect. We're dealing with now, these are 10 different human samples that reflect the kind of heterogeneity of disease and genetics and environment that we know characterizes analysis of human samples compared to the mice, which are genetically inbred, perfectly identical conditions, microbiome, et cetera. And so we think this is relevant to humans as well. Now, what was especially satisfying about this model was that, or at least the idea that there's a growing fraction of interactions that are autocrine with progressive fibrosis. What was satisfying is that in part by coincidence and in part by searching, we connected to Miri Adler, who was a visiting Fulbright investigator from Israel, who was working with Ruslan Mezatov and Uri Alon. And as it turns out, 2 years earlier, they had published this model that was based strictly on theoretical considerations, where they, and Ruslan in particular has been working at this for a number of years, they defined a bistable circuit between myofibroblasts and macrophages. And they defined two ultimate states, and this wasn't restricted to liver. This was a sort of a global fibrosis concept. And they predicted that there would be hot fibrosis, particularly early on, where you have mostly macrophages, and the hot refers to the inflammation or the infiltration of inflammatory cells, A similar analogy, which is used more commonly, is cancer, where you have hot tumors with infiltration of immune cells. And so they defined a predicted state in which there would be hot fibrosis, where you have macrophages co-populating the tissue with stellate cells. I'm sorry, macrophages are in purple, co-populating the system with stellate cells in orange. And then this would eventuate or evolve over time to become a myofibroblast-rich scar with less, if any, immune cells. This is particularly satisfying to those who think about NASH, because there are, in fact, two broad states. We have inflammatory NASH, which is depicted here, in which there's a lot of immune cells, certainly a lot of stellate cell activation and scar accumulation. But we also see this so-called burnt out MASH cirrhosis, where there are dense septi, but relative paucity, not a complete absence, but a relative paucity of immune cells. And so it suggests that the disease evolves, and as I'll tell you in a minute, so do the therapeutic targets associated with this fibrosis, these fibrosis stages. So just to summarize this, they predicted that cold fibrosis would be fueled by hepatic stellate cell autocrine signaling mediated through their physical interactions. So again, it starts with a physical interactome with dense network that evolves, and it's associated with functional differences. And some of those functional differences I'll point out now. If I can move the slide. So we generated, or Sammy generated, a list of overall of about 60 different receptor ligand interactions that were predicted to drive this autocrine signaling. Among those, we took out 51 that seemed to overlap between the human and the mouse data. Those obviously were more relevant so that we could model them in mouse and know that they had some value in humans. And so we selected NTF3, NTRK3, which is neurotrophin factor 3 and its cognate receptor. And in fact, confirmed that in stellate cells of NASH patients and in NASH mice, the receptor and the ligand were co-expressed. That was true also in humans. NTRK3 and NTF3 both co-expressed by activated stellate cells. Sammy also used immunocytochemistry to confirm the presence of the NTRK3 protein overlapped with alpha smooth muscle actin, and this is the overlays, just both in human and in the fat MASH model, confirming that both the ligand and receptor are present in the same cell that could account for both endocrine as well as autocrine interactions by this pathway. Now, fortunately, one of the reasons we chose NTF3, NTRK3 was because very little was known about it in stellate cells. Stellate cells. Another reason we chose this pathway to interrogate is because there is a clinically tested asset known as LOXO 195 developed by Bayer that is currently in clinical trials for tumors characterized by NTRK3 gene multiplication or duplication. And so we were able to obtain the drug, and we performed a pretty severe study, meaning we gave the animals 24 weeks of the fat NASH mice. This is what they look like: some steatosis, lots of bridging fibrosis and nodules. After only 4 weeks of the NTRK3 antagonist LOXO 195, there was a dramatic, albeit not complete, reduction in fibrosis. And this is quantified as in the bar graphs shown here, and this is the raw data that generated these bar graphs. So there was, again, a marked decrease in fibrosis, collagen, morphometry, and architecture characteristic of cirrhosis. And so this really has been very satisfying, because it suggests that the targets associated with stellate cell activation are not static, that they change with the progression of disease. So whereas earlier stages of disease, there's presumably more crosstalk between immune cells, stellate cells, and maybe endothelial cells, whereas at the late stages of diseases, a whole new panoply of targets appears that are driving this autocrine signaling. It also suggests that the drugs that are being developed and tested in NASH to mitigate the metabolic features may have no impact on this advanced stage of disease, because there are no longer major metabolic pathways driving the fibrosis, but rather there is now autocrine interactions that need to be interrupted directly if we're going to have a chance to move the needle on cirrhosis therapy. Speaking of that, I want to just introduce in the last few minutes some approaches that we're following and also pursuing. specifically cell therapy of fibrosis and senescence of stellate cells. So just to return to this paradigm, I emphasized before that senescence has been predicted as a potential driver of hepatocellular carcinoma for a number of years. And just to remind you, senescence is basically a result of cellular damage, stress, or replicative exhaustion. There is a unique cellular secretory phenotype associated with senescent cells known as SASP. And these are the features here, cytokines, chemokines. And of course, this has an impact on the surrounding stroma, particularly cells, both hepatocytes and liver and immune cells. And these cells can be cleared through an immune response. But in general, it's thought that accumulation of senescent cells is part and parcel of chronic tissue damage. These are the different biomarkers, and the list is growing. And the features of senescence in each cell tend to be relatively unique, meaning the combination of canonical markers seems to be very unique. It's not the same 4 or 5 markers in every cell or every tissue. One of the best known is actually beta-galactosidase activity that recognizes SASP, and it's important to distinguish the beta-galactosidase activity that's associated with lysosomal activity, which is detectable at a pH of 4. And here we're talking only about beta-galactosidase activity detectable at a pH of 6. And so in the studies I'll show you, and in most of the literature, that's what they're referring to when they talk about beta-galactosidase activity. One of the studies that suggested that the senescence associated secretory phenotype was published in in Nature by Yoshimoto in 2013. And this is just a diagram sort of summarizing that work that was written by Nick Shackle later that year in Hepatology. And the basic construct is that a high-fat diet and obesity lead to a change in the microbiome. I'm on thin ice here because Barbara Rehrman is listening, and I'm a deep admirer of her fantastic work with the microbiome. But nonetheless, these investigators suggested that there's an enterohepatic circulation between the bowel-derived abnormal microbiome features and stellate cells that lead to the emergence of a SASP, which is characterized by many features, including IL-1 beta. And so it suggests that this contributes to a tumor-prone environment. So this led us to ask in general, and then more specifically, what if we could deplete stellate cells? First of all, what if we could deplete all stellate cells? So the expectation, and in fact, the reality, as we learned, is you would have a loss of many of these canonical features. And we actually were fortunate to be working a floor away from a very talented geneticist and molecular biologist, Brian Brown, who's very, very creative. And he had created a system known as the JEDI system, which stands for Just EGF Death-Inducing T cells, in which he engineered mice to generate CD8 T cells that would only recognize green fluorescent protein as presented by MHC class 1. So the principle is you transgenically express GFP downstream of the promoter of choice. In this case, we used beta-P to GF-driven GFP. And then you administer the CD8 T cells, and they will seek and kill any of the GFP-expressing stellate cells. So this is what, this is work that really was driven by Youngmin Lee, as you'll see in a minute. She was a postdoc at the time and is now a funded faculty member at Vanderbilt. What you can see here is Desmond staining of a normal a mouse liver. And this again is the clearing method to show the Desmond beautifully wrapped around the sinusoids and co-stained with CD31 so you can visualize the endothelial cells and recognize that the pericytes or the stellate cells are wrapped around the endothelium. After administering the JEDI T cells, there was absolute and complete ablation of all Desmond positive cells, and I mean 99%. We had proposed in our grant to use this method to interrogate where the cells are repopulated and how does the liver get stellate cells back. The only problem was these animals were so sick, we couldn't keep them alive long enough to see if the stellate cells would repopulate. This initial observation was built into a beautiful story that Youngmin published just earlier this year, I think in Science Signaling in May, and really built on the NTF3 story almost by coincidence, where she proposed that normally NTF3 signals on hepatocytes, not through NTRK3, but NTRK2, which is also known as TRK beta. And this is particularly prominent in the mid-zonal region where we know hepatic repopulation is most dominant. And so under normal circumstances, NTF3 by stellate cells is preserving hepatocyte mass and proliferation in the midzonal region. What happens if you take out those stellate cells through Jedi T cells? Well, she started to see that the proliferation, the proliferative index of those livers, particularly in the midzone, was lost. And then over time saw really dramatic pathology where there was complete dropout and big sort of vascular holes, presumably because there was a progressive reduction in liver mass and mid-lobular hepatocyte dropout. So the moral of the story is it's great to think about depleting stellate cells, but one should be careful what one wishes for, because it's very clear, at least from these studies and others, that stellate cells also perform an important homeostatic role in supporting the mass of hepatocytes. So if one were so ambitious as to say, well, just get rid of all stellate cells, I think that's going to lead to severe problems in terms of hepatocyte regeneration and mass. On the other hand, there are subtypes, as we learned, of cells that may be more profibrogenic and more carcinogenic than others among stellate cells, and in particular, senescence. And so this is a study that was conceived and performed largely by Karina Amor and Scott Lowe's lab at Sloan Kettering. And they looped us in to help a little bit with the NASH modeling, but this is clearly their work. And they used an informatics-based approach to define what might be senescence-specific cell surface markers on stellate cells, on this subtype of stellate cells, and they identified urokinase plasminogen activator. What they did next, just to point out, again, we see that senescence based on beta-gal activity, surprisingly, is restricted mostly to desmin positive areas. That's shown more dramatically here in the 6-week fat MASH mice. So it looks like, number one, for reasons I can't completely explain, the beta-galactosidase activity, enzymatic activity, is largely restricted to desmin positive cells or stellate cells, thereby reinforcing the rationale for depleting senescent stellate cells. Now, to return to the paper from Christina Moore, they reckoned that if they could generate, along with Michelle Satellite, CAR T cells that would target any cell expressing UPAR, they could clear only senescent stellate cells and maybe yield a therapeutic benefit. And so these were CAR T cells targeting UPAR that depleted senescent stellate cells. And as they showed, they reversed CCL4 and fat-associated fibrosis. What you're looking at here is serious red staining in the absence of the CAR T. And after CAR T therapy, there's a dramatic reduction in the fibrotic area. There's also a dramatic reduction in the prevalence of beta-galactosidase expression, also going down. And perhaps most interestingly, and I would say reassuringly, albumin went up. Now, this could just be, I'm not sure what it just could be, but I believe that it could represent the impact of restoring hepatocyte health or the impact of reducing fibrosis and senescent stellate cells to restore hepatocyte health. Remember, there's this yin-yang relationship between fibrosis and regeneration. The more regeneration or pure regeneration does not mount a fibrotic response, and dense fibrosis inhibits regeneration. And if you think again about the Hep C story, how does a liver with advanced fibrosis, when treated only with a drug that targets the virus, how does that liver know how to regress scar? There's no treatment for stellate cells or even immune cells in that. It's strictly restoring hepatocytes to a healthy state. And that alone may elicit signals that we don't understand yet that mount or drive an antifibrotic architecture restoring program. So it's a lot of hand waving based on a small amount of data, but I will tell you, we repeated these studies and the rise in serum albumin is quite reproducible. Just to round out the story, we've also looked at UPAR expression in NASH. These studies and several of the other ones performed by an incredibly talented MSTP student. Her name is there and she goes by Yasha. She's returning to medical school, sadly for me, next July, but she's a talented investigator, and she showed that there's quite a bit of co-localization of UPAR and alpha smooth muscle actin. So the idea that stellate cells express UPAR, I think, is true in human cells as well. But we also learned that the expression of UPAR actually evolves over the course of experimental MASH, such that in early MASH at 6 weeks, most of the senescence staining is restricted to, and this is UPAR, and most of the UPAR expression is restricted to what look like stellate cells, whereas at 12 weeks of the model, now we begin to see more co-localization of the UPAR with, here UPAR with F480 cells or macrophages. So it looks like Scott Lowe's original study was correct based on the time points they used, where UPAR is restricted to stellate cells, but it won't surprise anyone who studies immune cells that eventually macrophages and presumably other immune cells begin to express UPAR as the disease advances. Now, one could say, well, this still showed a great therapeutic benefit, and it may well be that a UPAR-seeking CAR T has some relevance in fibrosis, even if it's not strictly targeting stellate cells. And that, of course, remains a possibility that a couple of companies are pursuing right now. Just to show a little bit more of a deeper dive, Yasha explored in much more detail the components of the senescence score for stellate cells, and these are shown here. These some have been classically described. And we can use this now as a sort of a marker for the subset of senescent stellate cells. Note that they're also non-proliferative. These are the quiescent cells here. These are the senescent cells. They've almost lost all Ki67 RNA. This is associated, the senescence pathways are associated with the senescence subtype, and some of the functional readouts are shown here. And what was really exciting is that Yasha was able to do RNA velocity analysis to effectively design, or I would say uncover, the sort of evolution of stellate cells during progressive injury. And this is shown here, where you see that we start with quiescent subgroups 3, 4, and 5, but the evolution goes to what's called a pre-activated cell, then ultimately an activated stellate cell, and from these activated stellate cells come senescent stellate cells. It's hard to remember the sequence I showed you, but this is exactly what we have been modeling, the idea that everything starts quiescent, goes to pre-quiescent, activated, and then a subset either deactivate, shown here, or go to senescence. And so it suggests that the phenotype of stellate cells may be quite plastic depending on the nature and the duration of the insult. And of course, we don't know yet, but are looking at how this pattern reverses itself, if at all, as fibrosis regresses after we stop hepatic injury. So just to summarize Yasha's work, we have shown that quiescent cells go to an activated precursor, then they are activated in the MASH, MASLD liver, and they can, two of the fates are either deactivation or senescence if the disease regresses. Put it another way, with respect to UPAR, most of the UPAR early on in the disease is expressed in stellate cells. As the disease advances, we've shown that myeloid-deprived suppressor cells and TREM2 macrophages begin to acquire UPAR expression as well. And it may well be that CAR Ts that deplete UPAR-expressing cells are also benefiting by depleting these cells as well. And then finally, Yasha's data, only a touch of which I've showed you, has defined a repertoire of targetable markers of senescent stellate cells that include the UPAR, but a number of other molecules, and in particular CD206, which is also known as MRC1. And we're developing, new kind of CAR T strategies to be more specific in depleting only senescent stellate cells. And hopefully I'll be able to talk more about that in a future lecture. One final example of the evolution of CAR T therapies is illustrated by this diagram. It's actually from a review I was honored to write to support a fantastic paper from Jonathan Epstein and his postdoc, Joel Rurik, which was published in Science last year. They said they had published a study earlier in Nature in which they showed that you could generate CAR T cells ex vivo to target a particular fibrogenic marker on cardiac myofibroblast known as FAP1, or fibroblast activated protein 1. They then reasoned, in part because they have the expertise of Carl June and Drew Weissman at Penn, that maybe they could actually administer a lipid nanoparticle that programs T cells not ex vivo, but in vivo to become CAR Ts with a specificity defined by the contents of the lipid nanoparticle. And that's exactly what they showed in this paper. They induced cardiac fibrosis with phenylephrine angiotensin. They then administer a FAP CAR, which is the CAR instructions, the cargo, in which the LNPs are coated with anti-CD5, so they target CD5 on lymphocytes. The LNPs are effectively processed through endosomal escape, and the mRNA is translated and released to generate a FAP CAR on the cell surface. in these mice expressing in vivo generated FAP-CARs, they were able to show that there was a marked reduction in activated fibroblasts, as they call them, reduced and improved ejection fraction functionally and improved diastolic function. It won't surprise you to learn that we're working with Jonathan now to see whether this system is relevant to human CAR Ts, but the concept is very exciting for two big reasons. Number one, if one can generate LNPs, as you know from the COVID LNP vaccines or mRNA vaccines, you can freeze a lipid nanoparticle and ship it anywhere you'd like as long as you keep it frozen. And that would be true of a lipid nanoparticle that can program T cells in vivo. So now you wouldn't be restricted only to a facility that has the technology to develop CAR T cells ex vivo. The second appeal of this is you're expressing an mRNA. So it's not integrating, it's not DNA, and so you can titrate and turn off the FAP-CAR activity. As you may know, some of the early CAR Ts that were administered for patients with lymphoma 10 years ago by the group at Penn, they have followed those patients who have a long-standing remission, and they still have the CAR Ts 10 years ago, and then maybe keeping their hematologic malignancies at bay. So exciting times in terms of cell therapy. How this will compete with conventional pharmacologic therapy remains to be seen, but certainly advancing the concept of CAR T outside of cancer into fibrotic indications as well. So with that, I'll close and just point out some concepts I tried to underscore. MASH is a rapidly rising cause of HCC, wherein cancer can arise before cirrhosis in about a third of patients. Single cell analyses have uncovered a tremendous heterogeneity of stellate cells that may explain some of the divergent studies exploring their role in hepatocellular carcinoma. The functional differences among these many different subsets are not entirely clarified. But of course, heterogeneity of stellate cells is also temporal. And as I showed you, the targets for early fibrosis based on stellate cell activation are going to be different than those for late stage fibrosis. And that may influence how we choose drugs based on the stage of disease, particularly in patients who are cirrhotic. And finally, the depletion of stellate cells is appealing for fibrosis and possibly HCC therapy, but it has to account for the fact that normal stellate cells support regeneration and liver homeostasis. So one has to be extremely careful and meticulous in only depleting disease-promoting stellate cells and not all stellate cells. So I hope this has given you a flavor of the excitement in the field, and I've been blessed by a fantastic group of young people in the lab that are shown here, great collaborators, not the least of whom was Scott Lowe, Ruslan, Miri, Uri. We had help from Neil Anderson, Matthew Pettyjean for the PharmaNest studies, and Jonathan Epstein for some studies I didn't show you, looking at the prospects for CAR therapy and liver fibrosis. So, and again, I'm blessed to have been funded now. Dr. Hoofnagel, my current R01 will take me directly to 40 years of R01 funding, which I'm intensely proud of. Some of this work, particularly by others, has been funded by the NCI, Department of Defense, and some industry contracts as well. So with that, I'll close and hopefully have some time for questions. Thank you again for the opportunity.
Thank you, Scott. That was amazing, truly amazing. I'm still kind of speechless about the CAR T cell therapy. We're trying to get this in solid cancers where it doesn't work, but it seems to be that maybe we're just using the wrong indication, and fibrosis is the way to go.
Early days, but you know, there's proof of principle.
Anyhow,
put it that
so
way.
are there any questions from the audience? Start your camera, speak up. Ben.
Scott, yeah. So fantastic talk. I have a question. Can you hear me okay?
Yeah,
yes.
Perfect.
Okay. You know, related to the macrophages and hepatic stellate cells activation fibrosis. So clearly there's, you know, some macrophage promoted resolution of a fibrosis. So then Stu Forbes did this human autologous macrophage transfusion to cirrhotic patients, try to treat the cirrhosis. Any follow-up or?
You're talking about basically macrophage, therapeutic macrophage infiltration?
Yeah, for the fibrosis, you know, treatment.
Yeah, so there's at least two companies, one of which I'm sure is, you know, publicly known as Resolution Therapies. This is based on the work of Stuart Forbes. They have a macrophage infusion. They haven't defined what those macrophages are for obvious reasons. but they are in the midst of clinical trials. What I can say is that it makes a lot of sense, and the company is still going. So I'm assuming things are going well, but I can't say more than that. But it is reaching the point of reality. One of the challenges has been, there's been a whole host of studies for 20 years administering stem cells or myogenic stem cells, mesenchymal stem cells. The problem is that every study characterizes their cell type that they infuse a little differently. And so it's been very difficult to standardize the phenotype of stem cells in a way that can give confidence that they're therapeutically beneficial. Macrophages, on the other hand, we know a lot more about. Their phenotype can be more robustly characterized and defined, and sometimes manipulated ex vivo. So I think this really makes a lot of sense. One of the things that it doesn't tell us, which vexes me no end, is that we still don't know where the proteases are that break down the scar, let's say, when we cure a patient with hepatitis C. What are the cells? What are the proteases? How are they regulated? Amazingly, we know it happens, but we still don't know. We had done studies when I was in Monty Bissell's lab 25 years ago, where we showed that stellate cells make MMP2, they make TIMPs, macrophages make MMP9, and somehow the field kind of, including us, moved on. And it's time to get back to the basic, blocking and tackling of uncovering proteases and their sources in the setting of fibrosis regression, because clearly the liver knows how to do this. If we can mimic what the liver does, we're going to have some therapeutic insight.
Okay, thank you.
Hi, Scott.
Hey, Jake, good to see you.
Great talk. Glad I listened in and hearing about all the exciting new development in your lab. So congratulations. So I have a general question. So you mentioned this a bit in your talk, that they are stellate-like cells in other organs, right? And these cells are highly plastics. So is there any evidence suggests that they may migrate from one organ to another, or trying to kind of influence something at a distance? Do you have any thought about that? Particularly with the hepatic stellate cells.
It makes some sense, but the evidence for a sizable fraction of stellate cells coming outside the liver is modest. As you know, or you may know, there is a cell type known as a fibrocyte. Tatiana Kiseleva has done the best work on this. And I think even by her accounting, it comprises no more than 5% of the source of stellate cells. Obviously, it's worth revisiting, particularly in systemic diseases where fibrosis occurs in more than one organ. For example, alpha-1 antitrypsin or scleroderma, things like that. I think the related question, Jake, is if you're treating activated stellate cells in liver, how do you know you're not going to do something bad to similar cells in lung or kidney? And that's a challenging issue. What I'd like to think is that where the injury is localized is where the amplification of these target cells is greatest. So if you have a patient with liver injury, they're not going to have activated myofibroblasts in their lung or kidney. And so by nature, if you're targeting a receptor that is activation specific, then you should avoid a lot of extra organ injury.
So you think there's less of a stem cell population for stellate cell? Most of them occur locally in terms of proliferation.
Yeah, that's the best I can tell you now. I'm not dogmatic about it by any means. The developmental source of stellate cells may provide some clue, and really great work done. Kinji Asahara at USC had papers that I thought very convincingly showed that stellate cells are derived embryologically from invagination from the liver capsule, so-called mesothelial cells. Mm-hmm. Whether that also represents a source of stellate cells during acute liver injury or a chronic liver injury, I don't know. As I mentioned, we had hoped, it was a big expectation of ours that we could define the source and the nature of repopulation of stellate cells, but we unfortunately picked a method that was far too efficient in killing all the stellate cells. So we never got repopulation, the animals died first.
Great, thanks.
Sure.
I see another hand.
Yes, it's a great talk, Dr. Friedman. This
Thanks.
is Kodo from Duke University. I have a question about stellate cell senescence. To my understanding, senescent stellate cells are less proliferative, less fibrogenic, So why like suppression, I mean, deletion of senescent stellate cell is anti-fibrotic?
Because we believe, or evidence would suggest, that senescent stellate cells have their own repertoire of inflammatory and perhaps injurious soluble signals. I mean, one of the things we're hoping to do is to actually capture these senescent stellate cells in models parallel to the ones where we deplete them, to really definitively characterize the contents and the transcriptome of truly senescent stellate cells, and maybe even culture them to establish what their secretory profile is and why they might be contributing to DNA damage or cell injury in some way. We don't know yet.
Thank you.
Okay, are there any other questions? It's 2:01, so I would like to thank Scott again. You know, this was an amazing lecture. I learned a lot, and like to thank everybody for listening and discussing. And this was the last lecture for this year, and Anu is probably going to tell us when we have the next one and who is going to speak again, because I don't know this. Anu, you're muted.
All right, our next lecture is on January 29th, and Lopa Mishra will be joining us speaking on that day. So you'll get announcements for that soon, and we have a full schedule for next spring in 2024. So we look forward to seeing everyone then.
All right.
This has been a
Thank
huge
you.
honor. I really appreciate it. And I have to congratulate you. These talks are fabulous. And we've been listening in on as many as we can. So thanks for spreading the word, Anu.
Wonderful. Thank you.
Really an honor. Thank you all. Happy New Year. And
Yes, Happy
Great
New Year.
talk,
Happy
Scott.
holidays.
Good to see you.
I'm going to turn around with another link for some of the folks.
Exactly.
Okay. See you shortly.